Good morning, everybody. Before I begin, let me just acknowledge the fun I've had working with all of these people. So, like the previous speaker, I'm one of the people that actually has had no personal association with Professor Khurana, but I did want to say that this conference has only strengthened my conviction that that's a big mistake. When I was 17, I made a deal with my parents not to go to college, and I spent 7 years reading and studying math, traveling, and working as an electrician. And one of the things that I did is I appealed to books on the history of science. That's really how I got started. And one of my favorite people was George Gamow, shown here. And I basically read many of his books. Probably some of you know about Mr. Tompkins and Wonderland, but he wrote quite a few other books that are more serious, such as 30 Years That Shook Physics, Biography of Physics, and so on. And this led me ultimately to learning about the RNA tie club. And from day one, basically, I spent 7 years kind of just learning about science. High school was meaningless to me. But learning about the cracking of the genetic code has been one of my lifelong obsessions. And it's really what Crick referred to as how the two great polymer languages are linked. So at any rate, it's a great honor to be here. I appreciate it very much. So the way my talk is going to go is that I'm going to start out with some musings on, a bit like in the last talk, about what does it mean to actually design life. And what I'm going to try to argue for is the idea that there's great virtue to enlightened empiricism, which is characteristic, for example, of the Bronze Age, if you think of materials. But there's also great virtue in having what you might call rational design, which I think characterizes the information age. I'm going to try and make an argument that the way that one reaches the status of being able to do rational design is by an interplay between quantitative theory and quantitative experiments. I'm going to use one particular case study, which has to do with the physical properties of DNA and how that impacts the readout of genetic information. What I'm going to try to do that's related to the theme of this symposium, and indeed this part of the symposium, is to try to explain to you my goal, which is that I'd like to be able to synthesize genes by equations. And I mean that in exactly the same sense that people synthesized the 777 on the computer. That was one of the first airplanes that was basically not tested in wind tunnels. And that's a great achievement, and I'll try and give you a sense of that. Then I'll describe the particulars, which is how to use statistical mechanics to try and reason out the behavior of, if you like, artificial genes. And then what I'll do is that these models lead to predictions that can be tested, so I'll walk you through our single molecule experiments and also single cell experiments aimed at doing what I would say is sort of concrete tests of these models. So at the time of his death, Richard Feynman's blackboard was photographed, and this was shown in Physics Today, and I don't know if you can read it in the back, but it's got all sorts of cool stuff on it. This is a list to himself of things that he wants to learn. And what he says in the upper left, which probably many of you have heard before, is what I cannot create, I do not understand. And I would say that yesterday we were, to some extent, blown away, at least I was, by the last talks having to do with basically creating artificial cells in different ways. And I think that this is a fantastic definition of what it means to design life. But I'd like to propose to you what you might call a more stringent version of what Feynman said, and that is what I cannot calculate, I do not understand. So that's really going to be the essence of what I'm going to try and walk you through. So let me start with this airplane analogy. So it's the 400th anniversary, probably many of you know, of Galileo turning his tube to the heavens, literally right now. And I recommend that you go read The Starry Messenger in celebration of that. But Galileo did lots of things. And one of the things that he did, which is shown in his two new sciences, is shown here, which is that he did what seems like the fairly lame experiment of taking a simple beam and hanging weights on it and asking how does it deflect. And the argument I'm going to try and make to you is that there's a direct line from this somewhat lame experiment to this very sophisticated and wonderful piece of technology. The point is, how can one synthesize a 777 on a computer? You have to know stuff. That's the simple answer. You have to know what happens when you hang a very heavy engine off of a wing. You have to know something about the deflection of this object in the presence of loads, such as in turbulence. Alan Mulally, who maybe some of you know is the current CEO of Ford Motor Company and was embarrassed recently by flying to Washington on a private jet, his career was really spent at Boeing, and he's the guy that was responsible for the building of the 777. And one of the things that I think about every time on one of these planes is he told me that they actually designed these wings so that we'd all be more comfortable in turbulence. Again, it's not accidental. They knew what they were doing. And so the same can be said about fluid mechanics. So here I'm showing you a diagram from Leonardo da Vinci. He thought about turbulence. People understand all sorts of stuff about flow over airfoils, and it's by virtue of starting at the beginning. Of course, that doesn't mean that you can't build a beautiful, beautiful cathedral without having the scientific underpinnings, but it means I think you cannot build a 777 in the absence of such underpinnings. So how does this fit into this symposium? So as I said before, what I would like to do, it's really in the spirit of the talk we just heard. I would like to be able to have somebody tell me the set of Lego pieces that they want to assemble as the regulatory apparatus for a given network, and I would like to be able to calculate what will be the level of gene expression as a function of the various tuning parameters that one might bring to bear on that problem. Number of repressors, number of activators, strengths of interactions, the bendability of the intervening DNA, loop formation. All these sorts of things are potential knobs that one can imagine dialing in. So what do I mean when I say knobs? Let's go back to the Galileo example. In thinking about the deflection of a beam, the things that I can do to this is I can say, well, let's change the length and examine the deflection. I can say, let's change the cross-section, let's change the shape, let's change the material that the thing's made of. All of those are possible experiments that one can do. You can change the load, you can change where you put the load, you can make a distribution of loads. All of those are tuning parameters, and one can then ask, do I understand how this thing works? And the answer in this case is absolutely yes. I understand it cold. Over here, if I think about a given regulatory network, then the sorts of questions I could ask, the knobs I might tune are number of repressors, the length of DNA loops, the sequence associated with the loops, the strengths of these operators, the strengths of these binding sites for the polymerase, and so on and so forth. So the point is that the level of transcription can, in principle, be dialed in by tuning all of these different parameters, and I would like to actually see, can I take Potashny's commentary seriously yesterday, take him at face value, try to calculate the sorts of things that he says when one varies these parameters that are implicated in these processes. So one of the things that I'm quite keen on is actually the idea that there's all sorts of fun ways of examining the fact that DNA is a physical object that matter to the readout of genetic information. I'm showing you 4 of my favorite examples here. So I'm going to emphasize DNA looping today, so I'll come back to that. The actual position of nucleosomes on genomes is not something that's random. And the idea is that if you give a histone octamer the choice of different sequences, because of the bendability of the DNA, some sequences are more favorable than others. Another question that's very interesting is if you have a target site, a protein binding site that's buried within the nucleosome, then you would like to know how is it that the accessibility of that site depends upon the burial depth. And then probably one of my all-time favorite examples, which is related to our speaker later today, Mike Levine, is when Steve Small was in his group, they basically worked out how the positions and, I should say, the presence or absence of these various binding sites is related to the laying down of the fly body plan. And here too, there's basically action at a distance. So what I want to do is just walk you through what in some sense might seem like a tired example, but at the same time I guess I'm going to make the argument that one of the useful things about model systems is that they're gifts that keep giving, in the sense that perhaps this is boring to biologists, but for me as somebody that wants to calculate how genomes work, I can go back to the simple question of lactose usage in bacteria, really the ideas that were articulated by Jacob and Monod, and try to figure out how does the probability that this particular gene is on as a function of various tuning parameters work. So I want to make an aside, which is I think that it's very interesting that when Jacob and Monod were thinking about these regulatory decisions, they hit upon 2 key examples, both of which involve DNA looping. And Professor Young yesterday talked about DNA looping. And what I wanted to show you is a beautiful experiment that was done in the group of Mueller-Hill. And there's other examples done even here at Wisconsin by Tom Record. What's shown here is the level of expression. Basically what's plotted on this axis is repression. So this is basically the reduction in expression as a function of the length of this DNA loop. And the point is you can see there's all sorts of beautiful features of this data, one of which is the periodicity. The periodicity is, at least in principle, easy to understand as follows. If I have a protein that's bound at 2 sites simultaneously, I now add 5 base pairs between those 2 sites, then what will happen is that the DNA will rotate by 180 degrees and you will now be out of twist registry. So if you like, this is a readout of the twist elasticity. It's also mysterious that the maximum in this curve is at 70.5 base pairs. I don't have time to go into that. But here's the proposition that I would like to put forth. And this, it's too bad that Mark's not here for me to say it to his face, but my strong opinion is that no amount of words and cartoons will ever suffice to describe this experiment. In other words, I would be embarrassed to go in front of Caltech undergrads and tell them that we understand this system unless I can explain the features of this graph. That's my opinion, and I'm going to stick with it. Okay. So I also wanted to note that this notion of DNA looping is not some backwater. So this is an example from Eric Davidson at Caltech. All that I want you to take away from this is that in a garden variety gene in sea urchin, here too, you see, they say there's 59, I think, distinct loops that are documented. So this DNA looping knob is a very interesting one. Okay, so what am I going to do now? I want to basically talk you through sort of a 3-pronged attack on this problem of transcription, just giving you a flavor for what it would mean to synthesize genes with equations. And then I will show you the kinds of predictions that come out of this, and then appeal to 2 different classes of experiments for examining these kinds of problems. So one of the ways that you could imagine trying to calculate how a gene behaves is using the so-called thermodynamic models. I don't offer this as the only way to do it, but it's a simple way that I think is appealing. The logic is very simple. I want to know the probability that RNA polymerase is bound to its promoter of interest. And for my purposes, that is a surrogate for what I mean by the gene is on. If polymerase is bound, for my purposes, that means that it is on. Interestingly, this is a competition between polymerase, for example, and repressors, which might bind here and occlude the binding site for polymerase and hence close it down for business. You might also, as Patashni told us last night, have activators that can recruit the polymerase. So how would you go about calculating the probability of polymerase being bound? Well, the way we calculate any probability is always the same. We construct a fraction. The numerator of the fraction is the statistical weights of all of the good stuff. The denominator is the statistical weights of all of the possible eventualities. So here, the numerator has all of the states that correspond to polymerase being present. The denominator has the empty state, polymerase bound, or repressor bound. And what I'm showing you here without offering you any details is that I know how to calculate the probabilities of each of those things, or alternatively, I know how to compute the statistical weights. So what comes out of that? I'm going to basically walk you through one set of predictions, although there's all sorts of different things that one can predict. So here's the idea. People have been arguing about the behavior of DNA using cyclization. And the idea is if I take a piece of DNA and ask for the probability that the 2 ends will be in close proximity, that will clearly depend upon the length of the DNA. Why? If the DNA is very long, by entropic considerations, it's very hard for those 2 ends to find each other. Similarly, if the DNA is very short, it's hard for those 2 ends to bend and find each other. So there's some middle ground where those 2 effects are sort of least severe. So you can measure, as shown here, the probability of cyclization, that's what's on this axis, as a function of the length of the DNA. This is a random DNA sequence. This is a nucleosomal positioning sequence. Now what I'm going to tell you is that I can use the free energy difference between these 2 guys to actually predict what the looping probability will be in a single molecule experiment, and I'm going to walk you through that. I can also predict what the level of repression will be. And as I say here, I'm all about dangerous predictions. When I say dangerous predictions, I mean that after the fact, you're not allowed to go back and say, oh, but I'm so sorry, I've got 12 more parameters that I'm going to now tune. I mean, put the head on the chopping block, and either you're right or you're wrong. And if you're wrong, you learn something. So that's the attitude. Okay, so now I want to walk you through the single molecule biophysics idea. So over here, what you see, that's pretty boring, but it's this experiment playing out. The idea was already told us by Sankar the other day. You take a microscope cover slip, about 1,000 base pair piece of DNA. You attach a bead on the end, and then what you do is you examine the fluctuations of this bead. And it's really rather simple. If this tether was attached to the table, then for a long tether I could wander around. If I had a short tether, I couldn't wander around very much. And by looking at the Brownian motion of the bead, you can actually probe the dynamics of the loop formation process, because when the repressor molecule binds at these two sites and makes this loop, this tether is shorter and the excursion of the bead is reduced. This is actually what the real data looks like, which is you have a bunch of snapshots of the bead position. But you can use those snapshots to actually deduce something much more rich, which is the RMS deviation. So this is a plot of how much the bead has jiggled off of the origin as a function of time. And what I want you to notice is that the red line corresponds to unlooped, because the tether is long, so the excursions are large. And then these two states correspond to loop states, and I don't have time to really go into why there would be more than one looped state, but suffice it to say that you can learn interesting things. So here, what I wanted to do now is just show you how you can dial in these different knobs that I've already told you I know how to control at the level of theory. So the idea, just to belabor the point, for reading these histograms, this is the number of events, this is the RMS, so this peak corresponds to the unlooped state, these two peaks correspond to the loop state. And what we're doing here is we're actually measuring, for example, how does the probability of loop formation depend upon the concentration of repressor. And there's an interesting effect here that at first might seem counterintuitive, which is that at very large concentrations, you get no loops. Why would that be? Because there's a repressor on each operator, and so there's no looping because you basically eliminated the looping effect. I bring this up because I think in the developmental biology setting, it might be interesting when you have all of these different binding sites. As cells divide, the concentrations of the various binding proteins can change. And as a result of this, you can get a concentration titration effect, which will lead to different loops being the favored loops. Over here, this is just to show you we can do this also for different lengths of the loop. This is a bunch of different choices in one base pair increments. You can see this is the unlooped state. Those are looped guys. The details don't really matter. How am I doing on time? Does anyone know? 3 minutes from being done? Okay, I'll skip this. And just say that I told you earlier that we had a prediction. This data is for a random sequence. The red data is for a nucleosomal positioning sequence, which we've taken from its usual lifestyle, which is in a eukaryote. We've installed it in the lac operon, and then we've used this experiment to measure the looping probability. And these curves are basically different choices for the free energy difference of looping between random sequences and nucleosomal positioning sequences. Okay, I just want to say a few words about how, using living cells, you can test exactly these same constructs, and also the power of actually having predictions which are not borne out. So what we've done is we've elected to use fluorescence as a way to read out the level of gene expression. It's very standard stuff. Basically, once again, I'm going to measure a ratio. In the numerator here, I've got the guys that have no repressors, and in the denominator, I have the guys that are looping. Notice the fluorescence here is much larger in these individual cells than in those. And by looking at the means, I can compute the repression. All of these things are the knobs that I referred to earlier that we can tune in our theory. And so what I wanted to say is that one of the things that you can see is that we have a prediction. Again, this is, I said at the beginning, I want to write equations that tell me how a given gene will behave. And the repression depends on the number of repressors and these binding energies. And this is basically two different choices of numbers of repressors. And these are different operator strengths, or different strengths of binding sites. The curves are what comes out of the theory. But I basically want to walk you through something which I think is very amusing. I've said that the level of expression depends on the number of repressors. That means it's very important to have a census of the cell. I need to know how many repressors there are. And I want to tell you about a really brilliant thing that Michael Elowitz, Yuri Alon, Peter Swain, John Rosenfeld, or Nitzan Rosenfeld and John Young thought of. It's the following idea. So first let me show you this movie. Oops. What do I do? I click on this thing? Okay. So what you're seeing basically is dividing E. coli cells. At t equals 0, they had some red proteins in them, and then what happened over time is that these cells divided and there was no more production of red. So what you're seeing is a dilution effect. And what I'm going to argue is that cleverly they figured out how you can measure the number of repressors by looking at fluctuations. So here's the thought experiment. Let's say I gave all of you flashlights, and I had photon counters, but I didn't know the calibration factor relating number of photons to number of people, but I wanted to count the people in the room. Then I ask you all to flip a coin. Everybody that got heads goes to the right. Everybody that got tails goes to the left. I measure the number of photons from each of the two groups, and I do that again and again and again. Well, on average, there should be n/2 of you in each group, but there'll be little fluctuations. And by looking at the fluctuations in the fluorescence intensity, I can actually back out, just by looking at the difference between these two daughters, I can back out a calibration curve that allows me to basically figure out the relationship between number of photons and number of people. Why is that important? Because once I know how to convert number of photons into number of repressors, I'm done. Okay, I really am literally almost done, so I will be the first. So let me just end then with the following comment, that we did this experiment on DNA looping in vivo now. I showed you earlier that there was a prediction, the prediction being that there should be a large difference in repression between random sequences and nucleosomal sequences. And when we actually do the in vivo experiment, we find that there's what seems to be like a sequence buffering in the in vivo setting, even though I showed you already that in vitro we find that there's this strong looping effect. So as Yogi Berra said, it's tough to make predictions, especially about the future. My closing remark is that it's useful, actually, when there's sort of this kind of conflict. And I think probably the greatest example of this that I can think of is in the 1800s, there was a law for the specific heats of crystalline solids, which is that they should all be 3R, where R is the gas constant. At high temperatures, that's fine. At low temperatures, it's a miserable failure. And there was no way out of it in classical physics. Lord Rayleigh said, what would appear to be wanted is some escape from the destructive simplicity of the general conclusion. And it really took Einstein and quantum theory to settle the deal. So my concluding thoughts are as follows, that I think it's interesting to try to go after this sort of rational design perspective on designing life, where I mean actually calculating things. And I think that this sort of tight interplay between models and experiments is necessary. I showed you, I hope, a little bit about how one would compute the behavior of networks. And to me, it's all about, at the end, making some predictions, which I think of as tuning these various knobs, such as the strengths of binding sites and so on. And what we found is that the experiment